welcome to Unit 2. The first chapter of the Unit 2 will be the Chapter 7, and we are starting with the Part 1 of Chapter 7. In the Part 1 of the Chapter 7, we are mainly going to discuss the principal facts and the theories of essentially the bacterial and the archaeal growth, and we are going to talk about some of the laboratory processes which involve these principal facts and theories in Part 2. So first, most of the bacteria and archaea reproduce by the binary fission. And this is a very simple strategy that usually you have just one cell. And this is an early phase of the cell cycle. And you can see the typical structure, cell wall, cell membrane. You have the unimolecular chromosome. Sometimes it could be multimolecular, but mostly unimolecular chromosomes. The ribosomes, and essentially what happens, the cell prepares for the division by enlarging the cell wall, plasma membrane, and also DNA replication occurs. And once the DNA replication is complete, at that time, the septum grows inward as the chromosome move towards the opposite ends of the wall, and other cytoplasmic components also distribute, and the septum is essentially synthesized completely through the center, and two daughter cells are produced. And all of these must replicate and segregate the genome prior to division. So this is essentially the binary fission, which is the general reproductive strategy of the bacteria and the archaea. So some of the bacteria reproduce by some of the different kinds of method, which is just not simple binary fission. Say, for an example, these Listeria monocytogenes that actually forms these bud-like structure. So this is not like an equal division that in a binary fission that we just saw. On the other hand, this is essentially seen in a cyanobacteria, which is Dermocarpa, and this is called essentially the biocytes. This biocytes is essentially multiple fission. So each one of them is a Dermocarpa, and this is the multiple fission. And here we are looking at a microscopic view of the streptomyces, and this is just a cartoon of that. So essentially a streptomyces spore essentially germinate, and it forms a germ tube, which can extend into this hyphae-like structure. And we call this as a branched hyphae. And there are some aerial hyphae can form. And on top of these aerial hyphae, the spores can form, and these spores actually disperse. So this structure is like the micellar structure of many of the fungus that we essentially see. But these are streptomyces. These are strictly bacteria. So these are essentially some of the odds of binary, other than binary fission. These are some of the odds. So in the next one, we are going to talk about the bacterial cell cycle, and they are divided into three phases. And it is a little bit different from the eukaryotic cell cycle. And we are going to talk about the three phases, understand the molecular mechanism of chromosome partitioning and cytokinesis, and understand the cytoskeletal filaments which are involved during this cytokinesis, and how the cell shape is essentially determined. So the first is that the cell cycle is a complete sequence of events which extend from the formation of a new cell to the next. And it contains three different periods. So the B period is when the cell is born, so birth. And at the end of the B period, we called this as the initiation period of the replication, or the C period. C for the chromosome replication, C period. And essentially it starts with the initiation. And as the chromosome is being replicated, the cell will elongate. And at the end of the C period, or the replication of the chromosome period, there's a termination of the chromosome. That means that the chromosome has been completely duplicated. And in the D period, what we essentially see is the nucleoid segregation actually happens. The two nucleoid, or the newly synthesized chromosomes, segregate to two poles, or towards the two compartments of the elongated cell, and essentially a divisome-like structure. We are going to see that, which carried out the cytokinesis, and during which the septum and the daughter cells are formed. This is the D period. So essentially why this is different, if you just can think about that this is the growth period, this is the synthesis period, And this is essentially the cytokinesis. So this is different from the eukaryotic cell cycle, where you usually see a G1 growth phase, then an S or the synthesis phase of the DNA replication, followed by a long G2 phase, and followed by the partitioning of, which is the mitosis, which is chromosome replication, and the chromosomes are segregated in the mitosis, followed by the cytokinesis. So this G2 phase is absent in here. So so the bacterial cell cycle is considerably different, essentially, from the eukaryotic cell cycle. So the most bacteria have a single circular chromosome, and they have this single origin of replication site at which the replication essentially begins. And this is known as the ORIC. So if you have a double-stranded chromosome, each of the chromosomes or each of the strands will have an origin of replication. And also it will have a terminus, which is the site which the replication is terminated, located at the opposite of the origin. And the replisome is the DNA synthesis machinery. And we are going to essentially talk about all the DNA synthesis enzyme in an entirely different chapter. Here, we are mainly concerned about how the chromosome is already synthesized and how it is being separated, not how the DNA replication essentially happens. That is a separate chapter. So the DNA replication proceeds in both directions from the origin, and the origin moves to the opposite end of the cell, and the rest of the chromosome actually follows. So how it happens? So here you can see that this is the origin of a replication, and this is the terminus. So the origin of, so this is the replisome, is essentially, these are all the different proteins which essentially are localized in that origin. And now you can see that these origin of replication, they are moving. We call this as the forks. These forks are moving down, and you can see that right here. This is the two chromosomes are being synthesized, and because the origins are now separated, there are two origins, and the two chromosomes are being formed. So that's how it goes, and detail of that we are going to talk about in a separate way. But the cell elongates as the chromosome replication and the partitioning process essentially happens. As it elongates, you can see the chromosomes separate, and the septation essentially is continuous. And the septum form begins with what is called, in the middle of the cell, a Z ring formation. And we are going to talk about detail about that. And these Z ring forms the septum, which divides the cell into a daughter cell. And you can see that two cells are essentially formed. This will one of the cell will undergo another division. This is a typical cell cycle that you see in the E. coli. So how this partitioning system of the chromosome work? So these work with a number of different proteins, and these are called the partition proteins, and these are par A, par B, and the par S is one of the region near the origin of the chromosome. So essentially, you can see that these are the two chromosomes that are now being replicated. So here is an origin of replication, and close to that there are some par S region, couple of thousand base pairs. This is certain sequences where the par B protein binds. Here there's an origin of replication, another one on the opposite side, and here also near the par S side, this par B binds to the par S side. So these proteins remain bound onto these, and we are going to see that there are several other proteins essentially happens, and this is very well studied in a kind of cell which is the Colobacter crescentus. And we are going to see that how these proteins, as well as this par A, separate the two daughters. So one in dark blue, one in purple, these mother and the daughter chromosomes, how they are separated. Remember that although we are saying mother and the daughter chromosome, the chromosome essentially have one original, which is the parental strand. One will be the daughter strand, because each one of them contains one new and one old, because DNA conservation, DNA replication is semi-conservative. So each one of them will have one of the older strand and each one of them will have, along with that, one newer strand. So it's essentially the same. So what happens that this is actually studied very well in Caulobacter crescentus, and very interesting bacteria. Matured bacteria is sessile with a stalk at one of the pole. Now, from here, when the bacteria divides, and after the bacterial division, this stalk in this area, flagella is developed, and these cells are called as swarmers, and they will swim away, and before replication, they will shed the flagella, and a stalk will be formed, and the bacteria will become sessile. And from that sessile bacteria, there will be another bacteria. It will form two cells, and one of the new cells will be another new swarmer, which will have the flagella. So this partitioning proteins have, or which essentially take the chromosomes in different compartments, or the two poles as well. So these, again, the par S site is close together near the origin of replication, and being early in the duplication, it directs the segregation of the two daughter chromosome, and par B proteins bind to the par S site and nearby. And one partition complex remain at the stalk of the pole. This is the stalk right here, whereas the other is guided by these par A proteins towards the other pole. So see that these par B proteins, they are pulled towards the other pole by this par A system, and it is a little bit more complicated. We are going to see that. So this is similar to the spindle formation. Just think about that. These par A sites are essentially the centromeres on the chromosome. So these, think about like eukaryotic centromeres. And from these eukaryotic centromeres, the two chromosomes are separated out and pulled towards the chromosome. So you can think about this par B and the par A system more works like a spindle by which the chromosomes are pulled towards the two pole. So we will discuss this a little bit more detail than is presented in a textbook, and this will essentially help it. So here what you are seeing is a Caulobacter crescentus, and see that this is a cell which is a swarmer. This does have a flagella. And this is the chromosome, and it is this lighter part is the chromosome. And what you're seeing that they are in coiled form. I mean, like it's a very large nucleoid, the chromosome, and it's in the coiled form. And at one end of the cell, near where you have the flagella, you have a area or a protein complex which is known as a POP-Z network right here, POP-Z network. And the other pole does have some proteins which is known as a tip N. And you can see that the nucleoid, these chromosomes are associated with this par A ATP dimer. So par A ATP essentially coats these nucleoids. Now what happens that this is essentially, of course, the DNA binding protein. Now what happens that the par B and the par S complex encounters the par A ATP when the replication begins. So essentially what happens that as the replication begins, this will be origin, this will be another origin separated. Of course, initially it started from here and separated out. And these are the par B and the par S complex, which is essentially par B, par S complex, par A, par B complex, close to the origin, close to the origin. The origin is just not shown. So what happens when the par S site and the par B protein interact with the par A dimers, these par A dimers dissociate essentially, and the ATP is hydrolyzed, and they become par A ADP, which is in this black circle. So these are par A ATP dimerized form, and when they are dissociated, they become par A ADP. And these are essentially depolymerization happens, and as the depolymerization happens, this essentially shrinks and pulls this par B and the chromosome towards the side. This is, you can think about the depolymerization of the microtubules as some of these motor proteins pull the chromosome towards the pole. It is not exactly the same, but the same to some extent is the same thing. So the free par A monomers, which are these black circles with black dots with the green circle, these are sequestered or moved to the poles by these tip N and the pop Z complexes. So this is this is right here is the tip N, and right here is a pop Z complex. The new pop Z complex formed here. The older pop Z complex is right here. Now, what happened? The tip N is recruited then at the divisional plane. So now you can see that two chromosomes are segregated. So the two chromosomes now reached the two compartments. And now what will happen? The tip N will be now recruited at the division plane to be left at the pole of the new daughter cell. And right here, you have the POP-Z network that has been formed close to this par B. This will be the new flagellar site for the flagella. This will be the swarmer daughter cell, and this is still attached with the stalk. So this was the swarmer cell. Now it is sessile with the stalk, and this is the newly formed cell, which will become swarmer. And when it goes into the division, it will again become sessile, and this cycle will go on. And the par A ATP structure form in the subsequent cycle of segregation, coating the DNA. And from here, it will the cycle will back again when the chromosome will start replicating. The par B complexes will depolymerize these par A ATP dimers and essentially pull the chromosomes towards the poles as well. And there's a detailed paper that you can see right here is the reference, and I have uploaded that paper as a supplementary paper for your own interest, but that much of detail is not necessary. The overview is good enough. So after that, we are looking at the cytokinesis. So this is essentially the C phase, or the chromosome the chromosome replication and segregation phase. The cytokinesis forms the two daughter cells following the cell division. And the septation is the word which you use, the formation of crosswall between the two daughter cells. And we are going to see that there is a selection of a site for the septum formation where it is going to form. Essentially, it's in the mid cell for most of the cells. And there will be an assembly of the Z ring, which is composed of the protein, which is an FTSZ, which is a cytoskeletal protein. And this will assemble also the cell wall synthetic machinery and the constriction of the cell wall septum formation. And we are going to look at the entire mechanism of this. So what happens is that the FTSZ protein is involved in that process, and FTSZ filaments are heterogeneous in size and widely distributed around the plasma membrane. And this is the first protein to localize in the future division site. And the FTS monomers essentially polymerize, which will essentially form a Z ring. But what happens that there is a system in the cell which is known as a min system, which contains individual proteins like min C, min D, min E proteins. And what happens is that this blue area, or the clouds that you see, that these are the min system or the min CDE proteins. What happens is this min CDE proteins oscillate between the two poles. It increases in here and then it increases in here. Why it is so? Because the min CDA system doesn't allow the FTSZ to polymerize. So what happens that if this area is high in min CDE, the FTSZ will not polymerize. It will try to polymerize here. So essentially, after some time, the concentration of min CDE increases on the other pole. So on this side, it will not polymerize. It will start to polymerize in this well. So this oscillation happens in each side and either pole. So as this oscillation of min CDE system happens, eventually as the cell elongates, this oscillation from the pole to pole allows the FTSZ to essentially come into the middle of the cell, and that will essentially start the septation process. This is the overview of the entire process. So the essentially the FTSZ polymerization must be coordinated with the timing, because if the too early, if the Z ring is constrict the cell, and if the nucleoid occlusion doesn't happen, or that means the newly formed chromosomes are not separated out, the chromosomes will be severed, and essentially it will not be distributed equally towards the two compartments. So this MIN-CDE system essentially coordinates that when this will happen. And there are some other proteins which are also involved. The SLMA, this codes the chromosome except at the replication termination or the TER region. And the Z ring can form when the SLMA tagged chromosome has moved away. So the FTSZ understand that when the termination site has moved away, only at that area, because the rest of the chromosome is covered by the SLMA protein, and at that time, the FTSZ ring, which forms a Z ring, can form. And this is formed by a structure which we call as a divisome. This divisome is the Z ring associated in the discontinuous and the mixed cell. So what you are seeing that right here, the divisome forms in the middle portion of the cell, and you are looking at the cross section. So remember that we are not looking at from this, but you are looking from this area. This is the cross section of the cell. So you can easily see that the outside, this is the divisome area where you have all the peptidoglycan, NAG and the NAM, which are alternating. Then you have the plasma membrane. Here you have the chromosome, and here you have the termination site, which essentially binds to some of the protein, which is, say, ZAP-A, ZAP-B. This interacts with this FTSZ, which start polymerizing around this divisome area. And if you carefully look at that FTSZ does have a dagger-like structure. It has a pointed end, and right here you have a barbed end. So what happens that FTSZ polymerization, how it happens, that they are going to be depolymerized from one end, and they are polymerized from the polymerization, or they're added this. We call this as a treadmilling, just as we see this in actin polymerization. So they are going to be depolymerized from this and added. And as this happens, think about that this structure is essentially increasing in many different places. It's just only one part, but there will be many of them. And that essentially will constrict that entire part, and the septation essentially form. And you can see that there are several adapter proteins with this like FTSA and the ZIP-A, and this anchors the FTSZ proteins, FTSZ polymer with the plasma membrane. And these are called the adapter proteins, anchor the membrane. So the Z ring is essentially the scaffold for synthesis for the cell envelope. And this uses the GTP hydrolysis is the energy source. Unlike ATP, the GTP is the source, and help of the GTP, this polymerization happens, and this will essentially constrict, because as the polymerization going on, there will be a pull, and due to the pulling, the plasma membrane will essentially come together from different sides, because this will be one, another will be here, another will be here, another will be here. All of them are going to work this way by pulling the plasma membrane towards the center, and the constriction essentially happens, and the divisome separate the two compartments this way. So here, what you are seeing, that after the cell is born, it enters into a growth where the single cell increase in size. So right here, you can see the coccus, and the coccus is dividing, And you can see that this is a new hemisphere that is formed. This is the old hemisphere. And the cell shape is usually strict and is passed through generation, but some microbes change the shape under certain circumstances. Say, for an example, a classic example is Helicobacter pylori. The Helicobacter pylori can be an infectious agent, pathogenic, and take its residence in the stomach lining or the stomach mucosa. And it is implicated in a gastric ulcer and in some of the cases, even gastric cancer as well in some chronic infections. Now, this Helicobacter pylori, they can be coccus shaped, they could be rod, bacillus shaped, they could be comma shaped, they can vary, but usually that is not the common way. Usually the cells are coccus, rod, or they are comma shaped. They have all these different structures. So what happens that in the coccus, as you can see, the division happens in one plane where you have the divisome forms the septum, and the new cell wall and the plasma membrane and the structure, everything is synthesized near that structure. On the other hand, you can see that in the rod, there are several bands of structure where the synthesis happens, and that elongates the cell. And this is essentially controlled by a protein which is known as MREB. This MREB, in the absence of MREB, this multiple areas, the MREB is similar to or a homologue of actin, as a eukaryotic actin, as a cytoskeletal protein. And MREB actually helps in these multiple areas of synthesis. In the coccus, this MREB is absent. On the other hand, that in a rod or bacillus, if you essentially, what you do, that if you do not have the MREB, they will essentially synthesize the new cell wall and the plasma membrane in just one plane. But when MREB is present, it is in multiple areas. On the other hand, there is a very interesting thing that is seen in the comma-shaped structures, which is a vibroid structure. This vibroid structure, the Colobacter crescentus, is also vibroid. It's also a little bit comma-shaped, but Vibrio cholera is very similar. So here you can see that the FTSZ and the MREB is present in here. In addition to those, that the curved structure or the curved side does have another protein called a crescentin. And there is unequal growth on this side. So the size, the crescentin, this area, the cell wall and the plasma membrane growth is less on this side compared to the other side. And that gives this unequal growth, gives the comma-shaped cells. So this is the overview of the different cellular growth, and this is how the different cell shape actually forms. Now, the peptidoglycan is critical in a determination of the shape, and of course, that how this peptidoglycan is synthesized. So the peptidoglycan can be synthesized in two different areas. So for the gram-positive bacteria, it is synthesized in the cytoplasm. And in gram-negative bacteria, it can be synthesized in the periplasmic space as well. So very interestingly, what happens that the NAG and the NAM, these are synthesized inside. So N-acetylmuramic acid and the N-acetylglucosamine synthesized inside the cytoplasm, and they are transported to the inside or the inner surface of the plasma membrane, and they're attached to these bactoprenols. These are membrane-bound lipids. They're associated with that. So here you can see a bactoprenol, which is associated with a NAM and the NAG subunits. Now this bactoprenol and the NAG-NAM unit, the entire part associates with another protein, which is known as a Mur-J flippase. These flippase work with the help of ATP, very, very energy-dependent process. And this entire NAG-NAM complex along the bactoprenol is flipped towards the outside surface of the cell. And this is also seen in a very interesting way in some of the eukaryotes. The eukaryotes, where some of the glycosylation of the proteins inside the endoplasmic reticulum happens, there is a anchor, lipid anchor known as a dolichol phosphate, which is associated with the membrane. On the outside of the dolichol phosphate, all these glycosylation moieties associate, and then the dolichol phosphate flips and bring them inside. Here it is just the opposite. So again, that is a glycosylation of protein, which is inside the cell, where the glycosylation agents are brought from outside, from the cytoplasm to inside the endoplasmic reticulum. Entirely different process. In eukaryotes, entirely different thing. But here, on the other hand, you can see that all these things are happening inside the cytoplasm, and then they are flipped through the membrane in a very energy dependent process with this flippase outside. And now there are different proteins, which are the glycosyl transferases, or the GTases, and transpeptidases right here, which essentially associate these NAG-NAM with the existing peptidoglycan layer. And sometimes what happens that to put these new NAG-NAM residues, sometimes you have to break these NAG-NAM residues in certain areas to essentially put the new structures in here. So here you can see that there are several different areas where the synthesis is taking place, and many of the places you have to essentially cut those NAG-NAM residues or cleave them to put the new subunits in between them. And this is essentially the process that goes on. And remember that this structure, which is the peptidoglycan structures, the cell wall structure, is essentially an armor-like structure, and this is also referred to as a saculus in your textbook. So the saculus is nothing but this armored structure, which is the peptidoglycan layer around these cells. So here you can see the cellular localization of peptidoglycan synthesis plays determine the role. The peptidoglycan can only form in the septal septum, and the FTS-Z localization is essentially involved in this process. Whereas what happens here in the elongosome, which is the rod complex, this happens with the MREB as a scaffold by creating filaments along the cytoplasmic face of the plasma membrane. And the growth occurs in under several bands around the cell, but not at the poles, but several bands along with the cell. And as we said, the same way the MREB elongosome complex recruit all these different NAG-NAM residues inside, and they flip them outside to be added. And here you can see in the carved structures, the crescentin localizes at one side of the cell, resulting in an asymmetric cell wall and the vibroid shape. On the other hand, the archaeal cell cycle, we are going to take a look at the sulfolobus, and they are more similar to a typical eukaryotic cell cycle. And they are different from the bacterial cell cycle. But still, compared to a bacterial cell cycle, very little is known about the archaeal cell cycle. So when we study the sulfolobus species, it indicates that they do have a growth phase, G1, followed by a dividend replication, which is the S, or the synthesis phase, and follows by a G2 phase, which is segregation of the chromosome, followed by the cytokinesis. This G2 phase was essentially not there, which is directly followed by the cytokinesis. So the growth, it was essentially the initial phase, which is the birth phase, or the B phase, followed by the C phase, which is basically the DNA replication and separation, followed by the cytokinesis. So that was in the bacteria, which is very different. And the segregation occurs with some of the segregation proteins like Seg-A, Seg-B protein system. This is similar to the bacterial partitioning system. Seg-A similar to the PAR-A. Seg-B is unique in archaea, but thought to function as a similar to as the PAR-B protein. But there is no PAR-S similar sites has not been found in the archaea so far. The archaeal cytokinesis, on the other hand, are similar to the eukaryotes, and these involve some of the protein like the cell division protein A, B, and the Cs. So the CDVA binds to the membrane and forms a non-contractile proteins at the mid cell, and it recruits the CDVB and the C and the FTSZs. And what happens, the CDBB, on the other hand, constricts and separate the two daughter cell, and this is similar to an eukaryotic escort 3 complexes. So these escort 3 complexes are endosomal sorting complex required for transport. These are a bunch of proteins. There are more of 30 protein complexes forms these escort 3 complexes, which form these multivesicular bodies. And these multivesicular bodies essentially bud out of the cell for different purposes. And even some of the viruses use this to get out of the cell, like HIV uses this to get out of the cell using this escort system. But essentially what happens that so think about that this is a cell and this is a bud, and this is essentially forming a ring contractile to cut this one off and separate out. That's basically the overview of that. The CDVC, on the other hand, recruited with the cell division B protein, is like the VSP4. And these are involved in the cutting off or the scission process, essentially. And you can see the Z ring associated with the new S layer, and that essentially separate out. Here you can see that these are the FTSZ is labeled, and the Z ring is essentially fluorescing in this archaea. So this is essentially the process of archaeal cytokinesis. Now you can see that the next part we are going to talk about the growth curve of the bacteria, and there are different phases, and we correlate with the changes and relate the growth constant to generation, doubling time, and we are going to go into the physical parameters of how the bacteria essentially grow. So the increase in cellular constituents that may result in the increase in the cell number, increase in the cell size, and growth, most commonly referred to as a population growth rather than the individual cell. So here we are going to talk about the growth of the population. So far, we have discussed the growth of an individual cell, how the DNA replicates and separates out. Here we are looking at the population level. So the microbial growth curve is seen when the microorganisms are cultivated in a liquid broth culture. And the batch culture is incubated in a closed vessel with a single batch of medium, and usually plotted as logs of cell number versus time. And essentially, this is a closed system. And as you can see that the typical growth curve of a bacteria contains a lag phase, an exponential phase. We also call this as a log phase, then a followed by a stationary phase, followed by a logarithmic declination or the death phase, followed by a bunch of long-term stationary phase, which can be from a few weeks to months to even years. So this is an overview of over time. This is the typical growth curve that we see with the bacteria. So now we are going to describe that at each phase what is happening in a little bit more detail. So in the 5 phases, the first phase is a lag phase. And at this time, the cell is synthesizing the new components, and it is essentially adapting to the new media and the other condition. And they replenish the spent material, ribosome, and making the ATPs. And eventually the cells replicate their DNA, increase in mass, and started to divide. So that lag phase, essentially, there is a slow growth in this phase. So the cells are just being prepared to be fully active. In the exponential phase, the rate and the growth division is constant and maximal, and the population is most uniform in terms of chemical and physical properties during this phase. And during the exponential phase, cells grow as quickly as they can form if the conditions available. And the final net growth increases with the initial amount of the limiting nutrient present. and the growth rate increases with nutrient concentration, but essentially it will saturate, it will not grow forever. So here you can see that when a cell goes into this exponential or the log phase of growth, as you increase the nutrient concentration, the yield of cells per milligram per ml, but essentially it will saturate. So that means the yield of cells will saturate. On the other hand, here you have the growth rate per hour, and as you increase the nutrient concentration, it will grow exponentially after a certain, and then it will plateau. Why it is so? Because what will happen that after some time, the cells will reach a condition which we call as a critical threshold. And the concentration of the cells, when they usually reach about 10 to the power 9 per ml, at that time, that is a critical threshold where the cells essentially don't yield much more. And also what happens that when you increase the nutrient concentration, as the new cells are being formed, on the surface of the cell, on the on their plasma membranes, on their lipopolysaccharide layer, if they are gram-negative bacteria, through their cell wall on their plasma membrane, there are all these different transporters for the nutrients, which transports the which transports the amino acids as well as all the sugars that are required for their growth. And at a high concentration, at a certain way, that all the cells which are being produced, all of their transport system is completely saturated. That means 100% of their, at the stationary phase, 100% of their nutrient transport system is being utilized. It cannot go any farther. And that's the reason that we see this saturation kinetics at one point of time. But essentially, as we can see, that as long as we keep the nutrients available to a certain limit and do not replenish the nutrient, it will go. And finally, what will happen that in a batch culture, which is a closed system essentially, the growth will eventually cease. So we will not supply any material in here, and the total number of the viable cells will remain constant. And in the stationary phase, the balance between the cell division and the cell death happens. So that means the number of cells that are being produced is equal to the number of cells pretty much which are dying. And the population may cease to divide, but at this time they may remain as a metabolically active. And why we reach the stationary phase in a closed system? Because remember that the nutrient limitation and also limited oxygen availability, because oxygen is not too much soluble in the liquid media. So even when we use the agitation or oxygenation, still there is limited oxygen availability, and gradually there will be toxic waste accumulation. And as we said, that if there's a critical population density is reached, essentially the cells will cease dividing, and that is observed in the stationary phase. Then what happens that in the death phase, the number of viable cells decline exponentially, with the cells dying at a constant rate and of course faster. And is this time all the nutrients are deprived, that means taken up, and buildup of toxic waste cause the irreparable harm to the cells, and they basically die. But after this death phase, there is a long-term stationary phase where the bacterial population will continually evolve. And this process marked by successive waves of genetically distinct variants, and natural selection may occur within a single culture. And these long-term stationary phase, a very few number of viable bacteria may be present in the culture, which may be there from few weeks to months, even years, depending upon what kind of bacterial genus and species it is. So the next part that what we are looking into is the mathematics of the growth, and we are just going to understand just the very basics of it. And again, we are not going to do too much of the math, or not required for the scope of this course. So essentially what we are looking, first we are introducing a word which is a generation time, which is the time doubling time. this is a time for required population to double in size. So basically what happens that one bacteria divides into two, and the generation of the doubling time, suppose this is 20 minutes. So generation 0 to 1 is 20 minutes. So you can see that the number of cells from 20 to 40, 40 to 60, 40 to 80 is will go 2, 3, 4 as generation, and it will go to 2 to 4, 4 to 8, 4 to 8 to 16, and so on. So essentially, if we plot these with the time, here you have the number of the cells right here, and this is essentially the plotted against the number of the cells. On the other hand, what you can see that right here, on the other hand, this is the log number of cells. So if you just plot it against a log scale, as you can see right here, you can see that this growth is exponential. So 2 to 4, 4 to 8, 8 to 16 over the period of time. And this is your log plot right here. The growth is logarithmic. And that's the reason that we essentially say that this is the exponential growth. So how we calculate the growth rate constant, or K, number of generations per unit time. So here there are a couple of things that we are looking at. The N0 is the initial population number. So this is the N0, and this is the number of cells in the culture that we are essentially seeing is 2 to the power n. Nt is the population at the time t, and n is the number of generation in time t. So for the population reproducing by the binary fission, the Nt, the population of time t, is N0, the initial population number, multiplied by the number generation of the time, 2 to the power n, because it's binary fission, 2 to the power n. And if you solve this, n, solving for n, the number of generation, when all logs are base 10, so log nt is log n0 plus n log 2. So if you just look at these, solve this, basically, you are going to get k is equal to n over t, and which is equal essentially the log nt minus log n0 over 0.301 t, which is essentially the number of cell in is right here. So right here, 0.301, as you can see, that is your log 2. So here we are looking at the log 2. So, but that's 0.301. So once you have that, you can see that this is essentially gives you the idea, the growth rate constant. So this is your growth rate constant. And let's take a look at this. So now we are calculating the generation or the doubling time. If a population doubles, then NT is 2N0. And if you substitute the N0 into the growth rate constant, the equation solves. So K is equal to log this essentially, and K becomes 1 over G. So on the other hand, the generation time, G, is reciprocal to the growth rate constant. So just these two take-home messages are important. So remember that the growth rate constants in 1 over generation time, or the generation time is 1 over K. So the generation time and the growth rate constants are a reciprocal relationship. All you need is essentially this. These detailed calculations are not necessary. So here what we are looking at right here, this is the time, this is the number of cells, and here you are looking at the lag phase, whereas this is where the exponential phase, and when we are doing the mathematics, we are considering just this portion essentially. And right here, you can see that right here, the number of cells, 0.5 into 10 to the power 7, here 1 into 10 to the power 7, and this is the doubling, and this is one generation, this is another generation. And so this is the generation. And this is generation in between this. This is essentially 1 over K, or the growth rate constant. So this is basically the overview of that. Again, we are not going to go into the detail of any mathematical expression. Just if you know the very basics, that what it is, the relationship of the growth rate constant with the generation time, that is good enough. So here you can see or appreciate the generation times of many different bacteria and archaea and eukaryotes. You can see that as 0.35 hours, as less as, say for an example, 20-25 minutes, to even Treponema pallidum 33 hours, some of the archaea 45 hours, and you can see that it varies a lot in different incubation temperatures all across the spectrum. So after that, we are going into a section where we are looking at the environmental factors that affect the microbial growth. And we are looking at the different adaptations of the extremophiles, acclimate changes to their environment, how they do that. We are looking at how the oxygen can form toxic chemicals inside the cell, and the enzymes, how they can protect them in their environment. So the most organisms grow in fairly moderate environmental conditions, and the extremophiles, on the other hand, essentially grow in harsh conditions that would kill most others. And when they grow, most of the organisms, they have a minimum growth. They have a minimum growth environmental factor, and then they have a maximal environmental factor and an optimum. And this minimum, optimum, and the maximum environmental factors are essentially known as the cardinal values for their growth, which could be the temperature as well. So depends. So all microbes must respond to the changes to their environment, and they all have this essentially optimal range of the environmental parameter for their best growth. So first, what we are looking at, the solutes affect the osmosis and the water activity, and what it is. When there is a change in osmotic concentration in the environment, it affects the microbial cell. So when the cells essentially are put into the hypotonic solution, at that time, what's going to happen? The lower solute concentration outside the cell than the inside the cell. Water enters the cells, but they may burst. But most of the bacteria contain the cell wall. The archaea contains the S layer, and is essentially doesn't happen. These cell wall components can withstand the turgor pressure. On the other hand, the hypertonic solution, the higher solute concentration outside the cell than the inside, the water leaves the cell and the membrane essentially shrinks, and they may separate from the inside of the cell wall. And these are some of the conditions that can happen in different kinds of environment. But most microbes live in a hypotonic environment, and they are protected by the cell wall and preventing overexpansion of the plasma membranes. And there are mechanisms to lower the solute concentration in the cytoplasm. And there are mechanosensitive channels in the plasma membrane which allow many of the solutes essentially to leave. And on the other hand, the protists use the contractile vacuoles to expel the water. So these are some of the ways that they can survive. So here we can see that the osmophiles, which are the halophiles, these halophiles require sodium chloride at a concentration about 0.2 molar. Extreme halophiles require salt concentration between 3 molar to 6.2 molar. Where do you find these? These you will find, say, Dead Seas. These you will find in the Salt Lake, in Utah. So there are salt mines. There are these extreme halophiles, and how they maintain these conditions. So here you can see that right here, this is a non-halophile. By close to 1 molar sodium chloride concentration, they will die. Whereas the moderate halophiles with 0.5 molar to about 3.5, halotolerant can tolerate up to 3 molar, whereas the extreme halophiles can grow from, say, optimum growth from 3 molar to excess of 6.2 molar. So this is essentially the sodium chloride concentration. So how essentially they do this process? So there are there are two processes by which they can survive. It's called the salt in and the salt out process. These are the adaptations. So in the salt in adaptations, they accumulate the potassium and the chloride in the cytoplasm, and some of the proteins need these very high salt levels. And there is also what happens in these conditions. Essentially, they survive. On the other hand, what happens that there is a process called salt out, and these keep the salt ions outside the cell, and they synthesize these compatible solutes that do not interfere with the growth. And these salt out compounds, they could synthesize like a choline, a betaine, then proline amino acids, glutamic acids, and the fungi or the protists sometimes synthesize sucrose, polyols like arabitol, glycerol, mannitol. These are different alcohols that is also present in some of the eukaryotes, and these are essentially the compatible solutes, and they keep. This is the essentially the salt out mechanism. And these two processes of the salt in and the salt out allow these halophiles to withstand these extreme conditions. So another very important factor in terms of these osmophiles and many other processes is the water activity. This measures the water activity, or AW measures the availability of water. Low water activity means most of the water is bound and not available to the microorganism. So this is 100 the relativity of a solution. So this is equal to the solution's vapor pressure to that of a pure water. So for distilled water, the water, essentially, this is 1. On the other hand, you can easily see the osmotolerant microorganisms that can grow over wide ranges of water activity, but optimally at higher levels. And Staphylococcus aureus is essentially one of them, because Staphylococcus aureus can even grow on our skin, and from the sweat and other, we do have a good amount of salt concentration on our skin, and the Staphylococcus can actually withstand that extra amount of salt. They are osmotolerant. On the other hand, the zero-tolerant microbes withstand high solute concentration, and these are found in, say, very dry area like a desert, or in desert where there is very little humidity in there, very dry environment. They can found in like a dry the dust particles, and also they are found in the preserved food, which do contain like a brine preserved food, or say vinegar, or very high sugar food, which are essentially the preserved food. They are essentially found in those conditions. So the next one, the factors, is a pH and relative acidity, or the alkalinity of the solution, and essentially is the negative logarithm of the hydrogen ion, or the H plus concentration. And again, we do not have to go into the basics of the pH, but essentially what are the different ones? The acidophiles growth best between pH 0 and 5.5. And here you can see that these are the acidic. Right here is the neutral pH, and these are the acidic pH, and see that these are the different organisms. So A stands for archaea, E for eukaryotes, and B for bacteria. So here you can see even the ferroplasma can grow at a pH 0, whereas this Acidonitrobacillus and Sulfolobus, which is they can grow in pH close to pH 2. On the other hand, Lactobacillus acidophilus or E. coli, they prefer just below the neutral pH. Staphylococcus aureus like to grow at a higher pH to just slightly alkaline, whereas these Alkalobacillus alkalophilus can grow pH 11 plus. So the alkaliphiles, or sometimes known as alkalophiles, grow best between 8 and 11.5. In contrast to the acidophiles and alkaliphiles, most bacteria protists are, on the other hand, neutrophiles, and they grow near close to the neutral pH. But a few of the fungi, they like to have a little bit more acidic surrounding. Some of the photosynthetic protists also favor slight acidity. And we can see that several archaeas are acidophiles. And also the alkalophiles are distributed in all three domains of life as well. The microorganisms respond to the external pH change by different mechanism and maintain a neutral cytoplasmic pH. So the neutrophile exchange the potassium for the proton or the H plus. So you can see acidophiles pump the proton H plus out of the cell, and the alkaliphiles exchange the internal sodium ions for the external protons. So there is a built-in mechanism to do this process as well. The next from the pH, we are for as an environmental factor, we are looking at the different temperatures. And the microbes cannot regulate their internal temperature, and the enzymes all have optimal temperature at which they function optimally. And below the optimum temperature, enzymes is not catalytic. And of course, a high temperature can inhibit enzyme function and even denature them, and they can destroy them. So the cardinal temperatures are essentially the minimum, maximum, and the optimal temperature, as we said. And across the different temperatures, depending upon that what range of temperatures the bacteria are growing, we divide them in different groups. So the psychrophiles, as you can see, they can grow in between 0 degrees to 20 degrees Celsius. These are the psychrophiles right here. Psychrotrophs, on the other hand, grow from 0 to 35 degrees Celsius, whereas the mesophiles are particularly optimum in between 20 to 45 degrees. Very interestingly, like all the pathogenic bacteria or that we encounter, all the pathogenic organism, they are all mesophiles, because that is essentially for particularly for the humans or for the mammals, the essential temperature 37.5 is essentially the temperature or close to in between say 37 and 40 for most of the mammals, and the mesophiles are the only one which are comfortable in that temperature range. Psychrophiles and psychrotolerance, or thermophiles and hyperthermophiles, are non-pathogens. Thermophiles, on the other hand, grows at 45 to 85 degrees Celsius. And think about very hot rocky areas, desert like Death Valleys, and hyperthermophiles, 85 to 100 plus degrees Celsius. These are found in many of the geothermal areas like mud pot, hot springs, and in the ocean floor near these hydrothermal vents where there are boiling water temperatures. And psychrophiles are, of course, found in the polar ice caps to even mountains and in the snows as well. And you can easily see that these are the different organisms. You can see this Chlamydomonas nivalis. This is an eukaryote. It is essentially a psychrophile. On the other hand, you can see that right here, the Pyrolobus fumeri, that can grow at a temperature of 130 degrees centigrade, whereas these Planococcus halocrypophilus can grow happily at minus 15 degrees Celsius. Whereas the Escherichia coli right here, you can see that the range of E. coli is essentially the mesophilic. And Saccharomyces cerevisiae, this is also, it can tolerate a little bit lower temperature, but its optimum is almost close to in between about 22 degrees Celsius is optimum for the Saccharomyces cerevisiae or the yeast. E. coli, 37 degrees is optimum growth temperature. So on the other hand, see that right here is the Trichomonas vaginalis. This is also a human pathogen, and the optimal growth temperature is around 37. So here you can easily understand that most of these Neisseria gonorrhoeae the same way. The Neisseria gonorrhoeae, E. coli, which is present in our colon, these are harmless, but there are some of the species of the rogue E. coli like O157, 87, those kind of E. coli. These can give you the bloody diarrhea and gastroenteritis. But no matter what, the E. coli, Neisseria gonorrhoeae, as well as the Trichomonas, these are all different human pathogen. They are all within the mesophilic temperature, whereas the Thermus aquaticus, Pyrolobus, Chlamydomonas, these are all beyond the range of a human body temperature or mammalian body temperature and are non-pathogen. So what kind of adaptations we essentially see for these bacteria or for the archaea. There are heat stable enzymes and protein synthesis systems that function at very high temperature. And protein structures are stabilized by a variety of means with hydrogen bonds and excess amount of proteins architecture, which are with the beta sheets. More of a beta sheets are essentially seen in some of these protein structure because beta sheets are held together with more hydrogen bonds than the alpha helices. So that's why more hydrogen bonding help to stabilize these structures. So the proteins which are more with beta sheets compared to the alpha helices are more stable at higher temperature. Also, these proteins contain these hydrophobic interiors where the hydrophobic amino acid side chains essentially interact with the other hydrophobic amino acid side chains. These are very stable interactions. And there are more proline. Proline is a very interesting amino acid which does have very rigid structure, and they can be formed in a cis and a trans form. And around the proline, they offer the rigidity of many of the proteins. And as the less flexible, they are less flexible in some of the regions, which are the key regions of many of the proteins, they do not denature that easily. And there are also many of the proteins which are the chaperone proteins, which also help to keep the proteins in a properly folded form. On the other hand, the membranes are stabilized by a variety of means. There are more saturated, more branched, and higher molecular weight fatty acids are present in the plasma membrane of the bacteria and the archaea, which can tolerate higher temperature. And we also know that many of the bacteria contain the ether linkage, and these ether linkages also can be covalently bonded, and they can form these monolayers, which are extremely stable at very higher temperature, which we see in the archaea. We have talked about that before. And many of them are resistant to hydrolysis, many of these structure at higher temperature as well. So these are some of the adaptation of the thermophiles. The next, we are going to consider the oxygen. Oxygen is present in the atmosphere, and depending upon the oxygen, it depends upon the microbe's metabolic process. So essentially, the oxygens are utilized in several processes, and at the electron transport chain, the terminal electron acceptor is used, and this terminal electron acceptor, when these different flavonoid electron transporter transfer their electrons to the oxygen, there is a condition at which there are some of these superoxide radicals are formed, which are O2 minus with unpaired electrons. So essentially, these will be detoxified, but short period of time, all of the cells which essentially live in the oxygenic environment or oxic environment will undergo this process. But other than that, there are some of the other influences. The effect of oxygen can generate these reactive oxygen species. Sometimes some of the photo or even the intense light can also deplete oxygen to form these species, these reactive oxygen species with unpaired electrons, and those could be detrimental from the cells. And depending upon that, we are going to see that how different organisms or the microbes tackle this problem. So there are 5 types of relationship to the oxygen. And this is what we are talking about is the molecular oxygen. So the obligate aerobes require oxygen. Like for us, like we are obligate aerobes. Without oxygen, these will be killed. This organism cannot survive without oxygen. Then there are also obligate anaerobes. That means that they usually killed in the presence of oxygen. They must be present in the environment which lacks oxygen. Presence of oxygen will kill them. There is also another group called microaerophile. They require oxygen, but requires about 2 to 10% of oxygen. This is just a little bit below the atmospheric concentration of oxygen. They require it, but a little less. Facultative anaerobes, on the other hand, do not require oxygen for growth, growth, but better in its presence. Because see that these are the best of both worlds. If they do not have the oxygen in the environment, they can respire by the anaerobic process. So they will just go in the glycolysis and they can just do the fermentation, and they will just only produce a little bit less amount of ATP per molecule of glucose. But in the presence of oxygen, they can follow the Krebs cycle and the electron transport chain, and they can produce far more number of ATP. So they will grow better in the presence of the oxygen. These are the facultative aerobe. Classic example is E. coli. E. coli grows very, very fast in the batch culture, that if we just culture them in the laboratory. But on the other hand, the growth of the E. coli are kept in check in the human colon because it is in an anoxic environment in the human colon, and they do not go very fast. And they are our microbial flora, and their growth is kept in check. On the other hand, the aerotolerant anaerobes grow with or without oxygen, and their growth doesn't necessarily better in presence of oxygen. So even if oxygen is present in the environment, aerotolerant anaerobes do not use it. This is the difference between the two. Facultative anaerobes will use oxygen if present, whereas aerotolerant anaerobes will not use oxygen even if they are present. So why these organisms are sensitive to oxygen and how these reactive oxygen species are formed. So the first one of them is a superoxide radical, which is a O2 minus unpaired electrons. Also, it can form from the O2 minus this hydrogen peroxide, H2O2 in the cell. This is also very, very toxic to the cell. And the hydroxyl radical OH minus, these are all very detrimental to the cell. So here you can easily see that these ROS or the hydrogen peroxide as well as the hydroxyl radicals, they will attack essentially the DNA, ribosome, periplasm, plasma membrane, plasmid, pili. Every part of the cell can be attacked with that, and these will essentially oxidize these. So these are all the reduced form of the oxygen, and essentially they will attack and oxidize these amino acids, the DNA molecules, and render them ineffective and will essentially destroy them. So in cellular environment, which essentially utilize oxygen for their respiration, constantly these superoxide radicals are essentially formed. Hydrogen peroxide and hydroxyl radicals are formed, and the cell constantly does have the process to detoxify them. These are your antioxidant system and enzymes which detoxify them. And we are going to see this essentially. So what happens that the microorganism must be able to protect itself from the reactive oxygen species. This is similar in the eukaryotics, much more robust system with many enzymes. And there are 3 enzymes which essentially protect this. So here, see what happens. The first one is a superoxide dismutase or SOD. 2 individual O2 minus with the proton, superoxide dismutase convert them to molecular oxygen, which is not toxic, and hydrogen peroxide. Now again, hydrogen peroxide is also toxic. So what happens, there is a second enzyme, which is a peroxidase, which with the help of NADH2, NADH2 is oxidized to NAD, and this O2 and hydrogen peroxide form 2 molecules of water, which is harmless. Another group another enzyme is the catalase. The catalase also use the 2 molecules of hydrogen peroxides right here with the O2, and it forms 2 molecules of water and molecular oxygen. This is your catalase enzyme. So you can easily see superoxide dismutase forms the hydrogen peroxide, which could be again detoxified either by peroxidase or by the catalase. So all these microorganisms does have superoxide, which can tolerate oxygen, must have superoxide dismutase and either catalase or peroxidase or both. So depending upon this, we can actually culture them in a different ways, the different bacteria experimentally, and we can see the different types of microbial growth in a test tube and understand that what their oxygen requirement is. And we also know that what kind of enzyme system they do have. So this is a typical test tube where you have a capped test tube, and this essentially contains a broth which is known as a thioglycolate broth. thioglycolate, thioglycolate broth. Or they also do have a solution of cysteine, which is the amino acid cysteine. So these essentially, the cysteine as well as the thioglycolate broth, these are oxygen scavengers. So once you essentially cap them, all the dissolved oxygen are going to be scavenged by the thioglycolate, and the whole part right here, the entire thioglycolate broth is going to be anoxic. But if there is a little bit of oxygen left at the empty space at the very top, a little bit of oxygen is going to be dispersed into this oxic zone. And depending upon this condition, we can inoculate the bacteria in this test tube and let them grow and look at their growth pattern. So if it's an obligate aerobe, they can only grow at the interface, which is near the oxic zone. If it is a microaerophile, they grow just a little below the oxic zone, because here this oxygen from this area, which is empty space, is diffusing, and the oxygen concentration is slightly less here below the oxic zone or the interface. Facultative anaerobe, they tend to grow better near the oxic zone, where they can carry out the Krebs cycle and the electron transport chain, and their growth is a little bit less towards the anoxic zone, where they cannot make that much of ATP and their growth is a little bit retarded. Whereas the aerotolerant anaerobe, they grow at the similar concentration at the entire zone, because doesn't matter whether they're in the oxic zone or anoxic zone, they do not utilize the oxygen and their growth is not dependent upon the presence of oxygen or not. Whereas the strict anaerobe tend to grow towards the bottom portion of the anoxic zone, because they cannot tolerate any dissolved oxygen from the environment, which is detrimental to them. So what kind of enzyme system they do have? So here you can see for the obligate aerobe, they do have the superoxide dismutase catalase peroxidase, all of the enzymes. Whereas the microaerophile may have SOD, they have peroxidase, and they may or may not have the catalase. Facultative aerobe, the same way, they have SOD, catalase, peroxidase, all of them. Whereas the aerotolerant anaerobes have the SOD and the peroxidase, but does not have the catalase. So you can see that all of these, which can grow in presence of oxygen, all of them must have SOD and either catalase or peroxidase, just one of them at least. They may have both, but at least one of them. On the other hand, here you can see with the strict anaerobes, the SOD, you do not have any strict anaerobes, you do not have any SOD, catalase, or peroxidase, so they do not have any detoxifying enzyme. So if the superoxide radicals are formed in these cells, there is no way the cells can actually survive, and the superoxide radicals will essentially kill the cells. So this can be done in the laboratory in this thioglycolate broth experiment. The next is the environmental factor, which is the pressure. The microbes that live on the land and the water live at a pressure of 1 atmosphere, and some archaea live deep sea with very high hydrostatic pressure. And these are barotolerant, adversely affected by increasing pressure, but not as severely as non-tolerant organisms. On the other hand, the piezophilic or the barophilic bacteria require high pressure for growth. And the change in the membrane fatty acid to adapt to this increasing pressure, and the lipid becomes more unsaturated and actually shorter. So these piezophilic or barophilic bacteria can be collected from the ocean floor, and if they are needed to be brought onto the surface You need special barophilic chamber or pressurized chamber to essentially handle these microorganisms, because if you expose these microorganisms to normal atmospheric pressure, they're instantly going to burst and you cannot culture them. Very difficult to culture them as such, these barophilic bacteria, but many have been isolated from depths of ocean floor. The next, we are looking at the electromagnetic spectrum, and here we are looking at the electromagnetic spectrum, which is the visible range, and they are lined by the infrared on the red side and the ultraviolet. And how the radiation affect the microorganisms. We can see that as we go the wavelength beyond the ultraviolet, we have the X-rays and the gamma rays. And these are some of the ionizing radiations which essentially can harm the growth of the microorganism. So ionizing radiations are the X-rays and the gamma rays, which can cause mutations essentially, and they can break the DNAs and can indirectly result in the death. And many times these gamma rays are utilized as a sterilizing agent in some of the industries. They disrupt the chemical structure of many molecules, break hydrogen bonds, and destroy their ring structures. And sometimes, on the other hand, we talked about this bacterial endospore, on the other hand, is Deinococcus radiurans, are extremely resistant to even ionizing radiations. And there are special chemicals that are essentially in association with their DNA, and their DNA is converted from the DNA B form to a DNA A form. And in that form, inside the endospore, their DNA remain very resistant to even ionizing radiations, very resistant to this. On the other hand, you can see the ultraviolet is a non-ionizing radiation, and most lethal wavelength is at 260 because it is absorbed by the DNA. And the DNA damage can be repaired by the several repair mechanism, although. So if they are damaged beyond a certain extent, you cannot. But a little bit of DNA damage, there are many systems that can be repaired, and we are going to talk about that later in this unit as well. Visible light, on the other hand, at high intensities generate the singlet oxygen, which is atomic oxygen, 1O2. So instead of O2, it's just O. And this is a powerful oxidizing agent. And the carotenoid pigment, which you find in carrots as well, these carotenoid group of pigments can protect many light-exposed microorganisms from what is called as a photooxidation, which is due to the singlet organism. So the next, what we are going to talk about, that how the microbes grow in the natural environment. We talked about them, that what are the different factors, but let's talk about that, how they survive, the microbes, how they survive the starvation, how in the presence of viable non-cultural cells in the food-water system might impact public health, distinguish sessile planktonic microbial lifestyle, and also the formation of biofilms and quorum sensing. We are going to talk about these in this factor. So essentially, the microbial growth are very complex and constantly changing, and expose microorganisms to overlapping gradients of nutrients, environmental growth, and essentially contain micro and the macroorganisms. And what are these? So here you can see that the few microbes live in a nutrient-rich eutrophic environment, which is abundant amount of nutrients. Most of them live in the oligotrophic environments where you have low nutrients. So essentially, the microbes have evolved many response to starvation and environmental stress. So because they are in an oligotrophic environment. So they have morphological changes, that is endospore in terms of starvation and critical condition. They can growth arrest, enter the stationary phase. They can use cellular components as the nutrients. Say, for example, they can ingest or digest the slime layer or the capsules if needed. And there are numerous proteins also help them to go through this mechanism or the starvation, or they survive in the starvation in an oligotrophic environment. And on the other hand, there are some conditions which are the viable but non-culturable state resume growth once the nutrition actually return. And there are also what we call as a persister kind of cells, which are also present in a nutrition deprived environment. So most of the microbes grow attached to surfaces which are sessile rather than free floating or the planktonic. But there are many bacteria which are also planktonic, and we find these in the seawater and in the water bodies. But what happens that when the microbes are sessile, these complex slime enclosed communities are called the biofilm and ubiquitous in nature in water and formed on any conditioned surface. Say, for example, you just take a rock from a pond or a lake. It feels slimy. That's essentially the a biofilm, which is essentially are very, very common in nature, which is a colony of many of these sessile microbial community. So how this biofilm is formed, the first, what happens, the substratum needs to be conditioned, and some of the molecules needed to be deposited, and usually these could be very simple as protein molecules. Say, for example, sometimes if we just think that in our tooth also, which can be used as a substratum for some of the mouth bacteria, in our saliva, we have many different proteins. And when these proteins actually coat the tooth, that is essentially the preconditioning molecule. And without that coat of protein, the bacteria will not be able to deposit onto them. So that's what the idea of what we mean by the preconditioning molecules. So the conditioned surface, the bacteria will essentially absorb, and sometimes they can leave the absorbed surface as well. So what happens that once they are on the surface, there will be a lot of cell-to-cell signaling, and they will try to secrete these exopolymers. So these exopolymers essentially will will help them essentially to grow exponentially. You can see, and they will secrete this polysaccharide matrix, which we call as the extracellular polymeric substance, or EPS. And the convective and diffusive transfer of O2 and nutrients take place. And essentially, many of these portions of these polysaccharide matrix, which is a polymeric substance matrix, EPS, they can be detached, eroded, or by sloughing, they can just move around into a different area and deposit it. So this is essentially how the biofilms are essentially formed. And a classic example for that, this will be the plaque formation on our tooth, or these can be formed on some of the medical devices as well. And sometimes it is dangerous when they are formed in medical devices like catheters, because once these bacteria are inside these polymeric substances deep inside, it is very difficult for many of the antibiotic to penetrate them, and it is difficult to actually get rid of these biofilms. So it is a dynamic community, and there is a heterogeneity in their activities, and there are many different types of interaction that helps. And also there are extracellular DNA that is released, and these DNAs are essentially extracellular DNA, eDNA. These DNAs are picked up by different cells, and they require they acquire new traits. And there are also extracellular RNA. There are these extracellular polymerase. These are secreted. These are EPS. And here you can see that there are several different processes, like you have adhesin molecules, which are the attachment factors, and there are flagella, there are pili. So many different structures of these microbes and extracellular polymers, they all essentially interact with each other. And use molecules to communicate, and DNA uptake can also happen in the community. And also, one of the very interesting way that there are properties which could not be predicted from studying a single cell, but some of the emergent properties of the biofilm microorganisms are only exhibited when they are in a biofilm community. And this physiological changes in EPS protects the microbes from the harmful agent like in ultraviolet and the antibiotics. And as we say, that antibiotic treatment fails, and some of the chunks of biofilm can be sloughed off, which can contaminate a drinking water system. Many of these water systems on their inside their pipe, you have these biofilm formation, and they can sometimes slough off. So the next is a cell-to-cell communication within the microbial population is known as this quorum sensing. And the bacterial cells can communicate via small molecules that diffuse in the environment. And in order to conduct business, a sufficient number of microbes must be present and participating. And this is seen, this type of activity in this bobtail squid, which does have a light organ. So during the daytime, they are buried in the sand, and in the nighttime, they essentially come out, and there's a light organ which can light them up. And this is essentially by a bacteria which is a Vibrio fishery, and this Vibrio fishery does use this this bioluminescence is provided by that, and a quorum sensing actually takes part in that. And we are going to talk about that how a molecule like in acyl homoserine lactone is essentially used by these structures. There are many different ways that these quorum sensing is responsible, because quorum sensing actually helps many of the pathogenic bacteria to release the toxin only after they reach a critical concentration. Because unless they reach a critical concentration, if they release the toxin, usually our immune system or a mammalian vertebrate immune system will kill those. But what happens that when a certain amount of bacterial growth occurs, and then when this bacterial population or a microbial population or the pathogen population release all the toxin at the same time after quorum sensing, that overwhelms the immune system, and we actually succumb to the disease. So the one of the system is this in acyl homoserine lactone, and this is what is called an autoinducer. And these normally moves across the plasma membrane from the cytoplasm. And when the cell population is low, you can see the autoinducers are synthesizing, population is growing in number, and at this time, it is not triggering anything. And at one point, these autoinducer molecule, when there are plenty of cell, it will reach a critical threshold. And at that time, these binds to its receptor, triggering a signaling network that initiate a cooperative process of doing something cooperatively. So on the other hand, say for example, see the Pseudomonas aeruginosa, as we said, that essentially use this, or say for example, Enterococcus faecalis, use these quorum sensing to express the virulence factor. The bioluminescent bacteria, Vibrio fishery, which is right here, the Vibrio fishery bioluminescent bacteria use this homoserine lactone when they for their light organs to produce this bioluminescence. And also, there are also several, N-acyl homoserine lactone is just one of the example, but there are also autoinducing peptides called the AIPs. These are present in the gram-positive bacteria, like Enterococcus faecalis does have these autoinducing peptides. And also, there are autoinducing short peptides. These are also, but along with that, there are some of the lipid-like structures that are also found. Say, for an example, in Candida, in fungi, the farnesic acid derived from this farnesyl moieties. These are lipid-like structures, and these are some of these auto-inducing structures. So there are many different types of molecules which can be used as the quorum sensing molecules or signaling molecules. And interestingly, sometimes what happens that in some of these biofilm community, there are interspecies communication can be formed by some of these molecules as well. That will be the end of the part 1 of the chapter 7, and this will be continued as the part 2 of the chapter 7.